Hello everyone, welcome to Mastermind. In this video, let us start a series on biotechnology. As we all know, biotechnology is important part of our general studies paper 3. So we have a separate section which is given on biotechnology in syllabus itself. So there is biotechnology, nanotechnology, etc. So in order to understand from the very basics of biotechnology, we'll start with this. The source will be NCERT itself. So if you look at NCERT class 12th, there is a section on biotechnology. And in that section, they have discussed about biotechnology in proper detail along with basics. So we'll start from there only and we'll proceed with that further. So if you look at the biotechnology chapter which is given in NCERT, then they are basically talking about principles and processes. Principles and processes in one chapter. So this is biotechnology, principles and processes. And in another chapter, they speak about biotechnology and its applications. Biotechnology and its applications. So let us start with principles and processes. So what exactly is biotechnology? So here the word itself is divided into two parts. First one is bio, which is life, and technology. Using this in technology. For what? All those things we'll be discussing. Especially biotechnology here, it deals with techniques of using live organisms. Or enzymes from these live organisms in order to produce the products. Or in order to help the humans. So that itself is biotechnology. So what is biotechnology? It deals with techniques of using live organisms or enzymes from organisms to produce products and processes useful for humans. Now, in that case, a lot of these are used in order to make, let's say, curd. We also make bread or wine. So these are also part of the process itself, correct? Because if we look at the process of curd, in that lactobacillus has been used. Lactobacillus, which is again bacteria, which is a live organism. You can consider it as live organisms or enzymes from live organisms. So this lactobacillus, which is a bacterium which is used in making curd from milk. Okay, so that can also be categorized as biotechnology. Okay, we will look at it. Next one is bread. In this, yeast are used. Okay, in this yeast is used to make bread because it is a microbe and that feeds on sugar, and in that fermentation process only, when there is more of carbon dioxide, because of that only the bread rises. So the reason the bread rises is because of carbon dioxide. And also in case of wine, yeast are used. So in this sense, curd, bread or wine, can they be categorized as a form of biotechnology? They can be, but here we are looking at larger section wherein technology is used. Here live organisms are used, but in the case of biotechnology, technology is used. That is, there is genetically modified organisms in order to achieve the same in a larger scale. Here everything is in a smaller scale. In a larger scale, we can take an example of in vitro fertilization. Now what is in vitro fertilization? Here again, there are two parts in this whole thing. First one is in vitro. What is in vitro? In vitro means inside a glass, and they are telling about fertilization over here. So whenever a reproduction occurs outside with the process of technology, that is in vitro fertilization we can say. But we'll look at what exactly is in vitro fertilization. In vitro fertilization, what happens is the eggs are taken from ovaries. Eggs are collected from ovaries, and then they are fertilized. These eggs are fertilized with the sperm. These eggs are fertilized with the sperm in a lab. In a lab. In this process, later on, a baby develops. So one full cycle of in vitro fertilization itself takes place up to 2 to 3 weeks. 2 to 3 weeks. Okay, so this is one full cycle of in vitro fertilization. That doesn't mean that the baby is delivered by 2 to 3 weeks. It is the process of fertilization which takes place up to 2 to 3 weeks. So this procedure is done to place one or more fertilized eggs, and then an embryo is produced, etc. So that is a process of in vitro fertilization. Now, because it is in a larger scale, it is a bigger model of this technology, where technology is being used with the help of live organisms. This can be called as biotechnology. This can be included under biotechnology. So the baby which is produced from this is called as test tube baby. This is called as test tube baby, because the word in vitro itself means that inside a glass. In vitro itself means inside a glass. Now this can be categorized under proper biotechnology. Now on this lines only, EFB. What is EFB? EFB stands for European Federation of Biotechnology. European Federation of Biotechnology. So they have given a definition of biotechnology that encompasses both the traditional view, which is this one, traditional view, along with that modern molecular biotechnology. Modern molecular biotechnology. So the definition which is given by EFB is the integration of natural science and organisms. The integration of natural science and organisms, cells, parts, and molecular analogs for products and services is called as biotechnology. That is what is biotechnology, which is said by EFB. What it is? The integration of natural science plus organisms. This may be organisms, or the organisms cells, or the parts of organisms, and molecular analogs with this, in order to get products or services is called as biotechnology. So I guess till here what is biotechnology is easier to understand. So we saw what exactly is biotechnology, wherein live organisms or enzymes from live organisms are used in order to get products for humans. Then we saw the example of curd, bread, wine. Here we saw bacteria, yeast, and we saw that the bread rises because of the presence of carbon dioxide. So all these smaller details are important for multiple choice questions. So sometimes it helps us in elimination of some options as well. Then we saw the proper categorization under biotechnology would be an example of in vitro fertilization. What is in vitro we have seen, and then eggs and ovaries, in order to collect the eggs from ovaries, and then fertilization takes place by sperm in a lab. That is the baby which comes out of it is test tube baby, and hence this is in vitro fertilization. Till then we saw what is European Federation of Biotechnology, giving a definition for biotechnology. Now let us move on to the principles. First one will be the principles. What are the principles of biotechnology? Now, there are two core techniques that enables this birth of modern biotechnology. There are two core techniques or principles. First one will be genetic engineering. First one is genetic engineering. The second one is bioprocess engineering. Bioprocess engineering. So these both have given birth to modern biotechnology. Modern biotechnology. First one is genetic engineering. The second one is bioprocess engineering. Now, what is genetic engineering? As the name itself says, it is about genes. It is about genes, correct? So, the modification of genetic material, modification of genetic material itself, that is DNA, RNA. So these modifications, so all these comes under genetic engineering. Then what is bioprocess engineering? This is the maintenance of sterile ambience in chemical engineering processes. For example, here there is the products like antibiotics, there is vaccines, enzymes, etc. So this is maintenance of sterile ambience in chemical engineering process. So in order to enable growth of only desired microbe. So we'll understand about what exactly is desired microbe, what is sterile ambience, what exactly are these modifications with examples. But I'm just giving you a basis for what we'll be discussing later on, because this is just the introduction video for biotechnology, right? So first one is genetic engineering. The principles, first one is genetic engineering. Second one we can understand about is bioprocess engineering. As there is a word process, you just mention it as sterile ambience. Sterile ambience. Okay. Genetic engineering, there is modification of genes. And in case of bioprocesses, the products will be antibiotic, vaccines, enzymes, etc. I hope till here it is very clear. In the next part, we'll discuss a little more about biotechnology and we'll complete with this biotechnology series. At the end of this e-paper, there are a few multiple choice questions for you to answer. You can attempt those. Thank you. I'll see you soon in the next one.